ERK-mediated mechanochemical waves direct collective cell polarization. Written by Naoya Hino, Leonie Rossetti, Ariadna Marin Yarado, Kazuhiro Aoki, Xavier Trepet, Michiaki Matsuda, and Sayoshi Hiroshima. Presented by Jennifer Arroyo, Bethel Andam, Nida Grayson, and Natalie Amirjanians. This study takes a look at how cell migration is able to be accomplished through ERK signaling and how that signaling can be maintained over long ranges. The hypothesis being explored is that clusters of cells accomplish long-range transmission of signaling for migration through a mechanochemical feedback system controlled by ERK activation through stretching and contraction of the cells with the use of directional cues. In order for us to appreciate the signaling occurring to accomplish migration, we need to first understand how this process is able to take place. Understanding and researching long-range transmission migration of cell clusters is important because this process is involved in many biologic processes such as tissue repair and embryonic development. Within these clusters of cells, direction of each cell is very important as it plays a role in the efficacy of intracellular ERK signaling throughout the cluster. The direction of the cells are managed by two factors: the leader cell's ability to sense direction from the environmental cues, and the follower cell's ability to obtain directional cues from the leader cells. In a cluster, the leader cells exist on the perimeter, and the follower cells are located behind them. Through this positioning, ERK waves are able to move through the cluster of cells, causing migration to occur opposite of the direction of the waves. The hypothesis is that this is able to occur through the stretch and contraction movement between the cells. When the cell is stretched, the ERK signaling in the cell is activated, which causes the cell to contract. This contraction motion causes connected neighboring cells to stretch and resulting in the same response. The long-range signaling domino effect through the cells is made possible by the cells being oriented in a front-rear polarity manner. This ensures that the signaling and subsequent migration occurs in one direction. The ERK signaling waves move in one direction while the migration of the cells move in the opposite. And here we have a visual representation of the process that was just explained and the hypothesis that will be tested. On the left-hand side, you can see there's a focus on a singular cell that is experiencing stretching, and so this causes the ERK signaling to become active, and then as a result, the cell contracts, and this process continues on a mechanochemical feedback loop. Which then causes ERK activation waves. As you can see on the right-hand side, here it depicts a cell stretching and ERK becoming activated and then the cell contracting. And then you can see the depiction right below. It shows the contraction of that initial cell causes the neighboring cell to stretch, which then activates ERK signaling in that cell, and so on and so forth. And so as you can see, this chain of ERK activation down these cells causes the ERK activation waves that will facilitate cell migration.
Bright images were obtained and processed under previous conditions to conclude time-lapse imaging. Immunofluorescence and confocal microscopy was when the MDCK cells were left in 4% paraformaldehyde in PBS for 15 minutes, permealized in 0.2 triton in PBS for 5 minutes, and then left to incubate for an hour at room temperature. Traction force microscopy is when B fluorescence captured the image with an inverted microscope, and then trypsinization took place in order to take the reference image. EGF stimulation assay is when the WT were seated in a 96-well plate and incubated for 24 hours, and then treated with different levels of EGF. Optogenetic ERK activation wave assay is applied when the migrating MDCK cells were treated with the EGFR inhibitor, which is then used to inhibit ERK activation waves and then measured. Timography was basically used as a mathematical measurement. So the values were averaged along the y-axis, and an intensity line was added along the x-axis, and then the lines were stacked along the y-axis to create points to help find our values.
In Figure 1a, to investigate the relationship between ERK activation and cell deformation during collective cell migration, ERK activity and cell deformation were evaluated by Freight imaging. As we've seen in Figure 1a and 1b, as the cells migrated toward free spaces, sustained ERK activation waves were propagated from the leader cells to the follower cells. The cell deformation, for instance, extensions and shrinkages were quantified by the cell strain rate in the direction of collective cell migration, which they called X strain rate. Similar to the ERK activity, the X strain rate exhibited repeated positive and negative values, namely extension and shrinkage, which propagated from the leader cells to the follower cells. To analyze the correlation between the ERK activation and the cell deformation, time series data of the ERK activity and those of the X strain rate were collected at the single cell level. Researchers then calculated ERK activation rate and the time derivative of ERK activity and X strain rate to evaluate these findings. They noticed that the ERK activation rate and the X strain rate oscillated almost together with an approximately 90-minute period. Furthermore, based on the Figure 1e, the temporal cross-correlation analysis revealed that the ERK activation rate lagged 3 minutes behind the X strain rate, indicating that the cell deformation waves precede the ERK activation waves. In other words, cells are first extended, followed by ERK activation, and then the cells start contracting.
Figure 2. ERK activation is triggered by passive cell expansion through EGFR signaling. Figure 2a and 2b, an experiment was done where MDCK cells plated on elastic silicone plate were stretched, and a comparison of ERK activity was done before and after the mechanical stretch. These figures showed ERK activation at 50% stretch of the MDCK cells. This indicates ERK is activated by passive expansion of cells. Figure 2c. MDCK cells were either treated with DMSO PD153035 or Mosostat at a stretch of 50% to test effects of ERK activation waves has on a collective cell migration. Both PD153035 and Mosostat is needed to suppress the stretch-induced ERK activation. Figures 2D through 2H, MDCK cells were stretched and lapsed at a time intervals of 1, 5, 10, 20, and 30 minutes. The cells lapsed were analyzed by M-blotting with either normalized fossilization levels of EGFR, RAF1, ERK half, and MEK half. M-blotting showed cells stretched, briefly increased autophosphorylated EGFR, and then phosphorylation of the downstream kinases. Figure 2 supports the hypothesis that ERK activation responded to different degrees of stretch and showed 30% stretch was enough to trigger ERK activation, which indicated degrees of stretch is proportional to ERK activation. The results of Figure 2D through 2H shows EGFR is activated by cell stretch and downstream signaling molecules. Figure 3. Cell contraction is triggered by ERK activation through Rho-associated kinase activation. Figure 3a. An experiment was done where seed cells with or without 2p-RAF expression was separated into two sections of silicone confinement. When removed, interface of the two cell populations formed. Figure 3b and Figure 3c, there was an interface shift near the side of the cell expressing the 2p-RAF, and applying MEK inhibitor and inhibiting the ERK activation caused suppression of the interface shift. Figures 3d and 3e showed that ROCK activity increased after the optogenetic ERK activation, then plateaued after the blue light exposure. Figure 3f and 3g, both ROCK and ERK activity displayed recurring unidirectional wave propagation from leader cells to follower cells. Figure 3H and 3I, ROCK activation waves and temporal oscillatory in each cell were eliminated by inhibiting ERK. Figures 3J and 3K, the oscillatory activity in each cell was eliminated and there was a decrease in basal activity of ROCK by inhibiting ROCK with Y27632. Figures 3L and 3M saw a decrease on the traction force applied on the 2P RAV expressing cells within 40 minutes after exposure to the blue light. Figure 3N, the traction force generation was reestablished by inhibiting ERK. Figures 3O and 3P, EGF activated ERK, which at the lateral end of the cell promoted F-actin localization. While inhibiting ERK produces stress fibers at the basal side of the cell. Figure 3 supports the hypothesis that ERK activity is only responsible for the oscillatory section of ROCK activity. The activation of ROCK is downstream of ERK, and is essential for ERK to activate cell contraction.
The major findings of this study is that the leader and follower cells both exert traction pressures on their substrates to propel cell movement during collective cell migration of the epithelial cells, resulting in long-distance coordinated cell migration. According to this study, ERK is a crucial protein controlling cellular mechanochemical feedback, which controls the sustained long-distance transmission of directional signals. The front-rear cell polarization is directed during the first phase of collective cell migration by the leader cells progress toward the open space, which exerts pulling pressure on the nearby follower cells. The pulling force strains the follower cells, activating the EGFR-ERK signaling cascade, which in turn activates the ROCK pathway to produce contractile forces at the backside of the follower cells. The next follower cell contracts, dragging the subsequent follower cells in a front-to-rear direction, creating room for cryptic lamellopodia to protrude for active migration. It is probable that Rho-GEF activation localized at the cell-to-cell interface contributes to the ERK-induced cell contraction via ROCK pathway. They further find that each cell has polarized rear contraction in response to ERK activation. The impact of ERK activation on the polarized contraction is explained by an antagonistic activation of RAC1 and Rho, because intracellular ERK activation may not be strongly polarized as a result of fast ERK diffusion throughout the cytoplasm. How mechanical inputs are transferred into signal transduction to activate ERK is a significant topic to study. Previous studies have shown that ADAM17 contributes to the spread of ERK activation in MDCK cells. This study discovered that stretching MDCK cells stimulates the EGFR-ERK pathway, as shown in Figure 2, and that this stretch-induced ERK activation necessitates the activity of the ADAM proteases. Researchers therefore come to the conclusion that EGFR activation caused by stretching includes the release of EGFR ligand via ADAM17. Future research will therefore be required to determine how the ligands in the ERK activation signal affect how mechanical stimuli activates the EGFR. Ethical issues in this study is that the shRNAs were introduced into MDCK cells by retroviral infection. Retroviral infection procedure can be problematic as it could infect humans. When a cell becomes infected with a retrovirus, the retroviral RNA is converted to retroviral DNA, which is then inserted into the DNA of the host cell. More retroviruses are then produced by the cell, infecting additional cells. Numerous diseases like cancer is linked to retrovirus. Limitations should be considered when utilizing retrovirus procedure in cell culture research, as possible mutations can cause novel diseases in humans.